Covance has a long history of working in this space, Covance being a global CRO. We have over a 20-year history of delivering on client needs in the cell and gene therapy space. That extends from our expertise, scientific expertise that cuts across from preclinical to clinical to post-market approval and long-term follow-up studies. It focuses on all elements of the precision medicine-based delivery of new therapies, gene replacement therapies, and adoptive T-cell therapies. Today, I'll be talking specifically about adoptive T-cell therapies, and it also relies upon our therapeutic area expertise in oncology, in rare disease, and other areas. We're able to coordinate those activities working with biotech companies and pharma companies, leveraging our scientific expertise, our regulatory expertise, as well as our commercial expertise when a new product comes to market. This is a focused area for us as a company. Covance is really focused in on the delivery of precision medicine, and cell-based therapies, gene-based therapies are the ultimate example of a personalized therapy. We've also focused on this through internal investments, but also partnerships with companies that allow us to look at things like single-cell genomics, minimal residual disease, et cetera, that are relevant for technologies and applications like adoptive cell therapy. As evidence of our expertise and our experience in this area, we have worked on all 3 of the approved CAR T-cell therapies, as well as both examples of gene replacement therapies that have been approved by the FDA. Biomarkers are relevant for adoptive cell therapy because of the complexity of adoptive cell therapy, as shown over in the right-hand graph, looking at biomarkers that predict response, that are relevant for toxicity, and also for relapse. The complexity is really highlighted by the fact that each patient is a unique case, looking at phenotype and genotype of the patient, but also each therapeutic approach involving an engineered T-cell is unique for that patient. So our ability to evaluate the CAR T-cells, the tumor, as well as the tumor microenvironment contribute to the challenge and the complexity of this area. There are a variety of biomarker considerations when we look at adoptive cell therapy. It starts with the phenotypic characterization of the engineered cells, the CAR T cells, the establishment of pharmacokinetic and pharmacodynamic relationships, the expansion and proliferation of the infused CAR T cells, the mechanistic markers of action and efficacy, as well as predictive biomarkers of response, or also that may identify the likelihood of relapse, resistance, or a safety event, an adverse event like a cytokine release syndrome. When we look at pharmacokinetic and pharmacodynamic measurements, there are a variety of things that are relevant. Starting on the left-hand side and working across this slide, again, we're looking at characterizing the proliferation, the expansion, and the persistence of the engineered cells. We want to look at the phenotypic characteristics of the CAR T cells. Is the antigen binding domain present, or has it been altered, for example? We want to look at immunogenicity, whether or not the T cells are eliciting their correct immunogenic response. We want to look at biomarkers of efficacy as well as biomarkers of toxicity. So examples of this would be persistence of the CAR T cells after infusion into the patient. This can be done by flow cytometry, measuring the phenotypic characteristics of the cells, or by using a genomic assay like qPCR. Immunogenicity can be measured by standard cell-based assays, immunoassays, and then looking at the efficacy of the action of the CAR T cell by looking at minimal residual disease. Is the target cell population being addressed and eliminated? And again, minimal residual disease can be identified and tracked either by flow cytometry or NGS, depending upon the nature of the tumor that's being addressed. Here's an example of using pharmacokinetic and pharmacodynamic biomarkers to look at the expansion and persistence of a CAR-19, a CAR T cell, that was evaluated in a clinical study. So here was a CAR T study in which the CD19 engineered molecule was used in therapy for young adults and children with acute lymphoblastic leukemia, ALL. The engineered molecule, the T cell, the CAR T cells were very effective in treating patients, particularly those with a low disease burden. And 90% of the patients showed complete remission at 6 to 12 months. Also, as shown in the right-hand graph, those patients that did respond best had a expansion and persistence of the CAR T cells as measured by a qPCR assay. Other things that we want to look at with regard to CAR T cells, we want to look at and characterize the cells themselves, the product, the drug, the engineered cells that will be reinfused back into the patient. We want to be able to detect the chimeric antigen in antigen receptor in blood by quantitative PCR, which is highly sensitive. We can also use flow cytometry to do likewise. We also want to look at tools that allow us to understand whether the CAR T cell that's been infused back into the patient activates the immune system. All along the bottom panel, we see examples of ELISpot assays or flow cytometry assays or other assays that allow us to look at the features of the engineered cells used in treatment of patients. We also want to understand something about the intrinsic T cell fitness of the CAR T cells or the engineered cells that are infused back into the patient. On the right-hand side of the slide, we see phenotypic characterization as we move from memory cells to effector cells, but also shown in the bottom right-hand graph are the phenotypic characteristics that are relevant as we move from memory to effector cells regarding proliferation, metabolism, etc. And understanding that is really gives us a clue, an insight into how effective the engineered T cells will be in addressing that patient's cancer. Safety biomarkers are relevant as well. We do know that there are a couple of safety considerations for adoptive cell therapy. CAR T cell therapies, while they are effective in a subset of patients, oftentimes a high subset of patients, they elicit a severe or significant immunological response, sometimes known as cytokine release syndrome. That's relevant to understand that because that can be treated with an anti-IL-6 receptor molecule like tocilizumab, first developed for autoimmune disease, but now used to tamp back an overactive immune response. We also want to look at the nature of the cells that are introduced and whether or not there are toxic, genotoxic elements relevant to the engineered cells. So is there replication competent retrovirus that is now relevant in patients, et cetera. So again, safety biomarkers are extremely important as well. Lastly, predictive or patient stratification biomarkers are relevant. As I mentioned early on, PK/PD biomarkers tell us something about both the persistence and expansion of the therapeutic agent, but also are they targeting effectively the tumor cells? So again, CD19 positive cells in ALL. Are we reducing that level, that tumor burden, down to an unmeasurable level? And that can again be looked at by genomic technologies or by flow cytometry. We also want to look at the nature of the immune response, and is it being driven effectively by the engineered T cells? And we also would like to look in many cases at in a global sense at what's happening in those patients, looking at a more comprehensive evaluation of RNA and DNA involving either qPCR, the nanostring IO360 panel, or RNA sequencing, looking at those elements that are expressed either in the tumor or in immune cells. So lastly, this area is a fascinating area. It really requires specific scientific expertise. In many cases, it requires a global partner to work with biotech and pharma to deliver on their adoptive cell therapeutic. So again, as I started out with, this area is very complex. It's unique in the fact that the patient is unique in its genotypic and his or her genotypic and phenotypic characteristics, but also the engineered cells used as a therapy are also unique within the immunologic milieu. And therefore, it's necessary in the assessment of any adoptive cell therapy to evaluate the tumor that we're targeting with the cell therapy, to evaluate the nature of the engineered and infused T cells, and to really understand how those interact in the tumor microenvironment. So it's been my pleasure today to talk with you about the role that Covance as a global CRO can play in biomarker development, biomarker strategy, and operational delivery of that biomarker strategy to support adoptive cell therapies. Thank you very much.